So in here, we can see how ATP molecule or adenosine triphosphate is used to store energy in the living organism. And energy, ATP molecules, it's usually referred as a currency or money in the living organism. So we will start with catabolic reactions. Catabolic reaction. So during catabolic reaction, complex molecules, for example, starch, proteins, lipids, will be breakdown to the simple molecules such as glucose, amino acids, glycerol, and fatty acids. And during this process, yes, energy will be released, produced. So this is what it's showing in here. Energy is produced. And during this process, adenosine D-phosphate and phosphorus will be connected and ATP molecule will be produced. During metabolism, also some energy which will be produced will be lost by the body and in form of the heat, and it will be lost into the environment. Next, we will talk about anabolic reactions. So during anabolic reactions, let's make it red in here, anabolic reaction. So what will happen? Simple molecules such as glucose, amino acid, glycerol, and fatty acids will be connected together, anabolic, yes, and will be produce more complex molecules such as starch, protein, and lipids. And again, during this reaction, usually ATP molecule is used because we need to connect molecules to produce more complex. So ATP molecules is used. It means it will be breakdown into the adenosine D-phosphate and phosphate. So, but during this process, some ATP molecules will be lost also in form of the heat. It will be released from the body and lost in the environment. Our next topic is enzymes. So first, enzymes are proteins that catalyze chemical reaction by lowering the activation energy. So overall function of the enzymes in the body to increase speed of the chemical reaction. And how they doing this? Because they're lowering activation energy. So let's see how this works. So we will talk about mechanism of action of enzymes, how it's called. It means we will talk about how enzymes perform their function, how they do what they're doing, yes? So in here, on the upper in here diagram, we will see enzyme. So in here, enzyme, so this is just molecule, some protein, and it have 3D shape, yes? And in here particularly, let's make it purple. This area of enzyme, it's called active site, active site. And this active site is site whereas different molecules which have exactly same shape like an active site will be attaching to the enzyme. And this molecule in chemical reactions called substrate. In here it's red. So it has this specialized shape exactly same as this enzyme have. So for each enzyme, we will have only one type of the molecules which have exactly same shape in here, and whereas they could attach to each other. And when enzyme and substrate attach to each other, like in here, they make enzyme substance complex. Now we have both this substrate and we will have enzymes that attach together in the complex. And mechanism how they attaching to each other called lock and key mechanism. It means that they both have this is perfect 3D shape, and only one substrate can fit in the active site of this one enzyme. It's very specific. Next, what will happen? During the next process in here, stage number 3, enzyme will change shape. How they call enzyme will change their conformation. So when enzyme is changing the shape, now what happening that during this process, substrate will be also changing the shape. And because of this, in this case, it's some connection between these atoms in the substrate in here will be broken. And then these two parts of the substrate will be produced. And in the next step, these two separate parts will be released. And now these two parts, they will call product. So we have originally substrate, then after they attach to the enzymes and make enzymes substance complex, next enzymes change shape, enzymes change their shape, and because of this, connections between atoms was broken in this substrate, and it was separated on two separate units, 1 and 2. So this is what in the end of the reaction, chemical reactions happening, two products was produced by this enzyme from substrate. And in the end, last stage of this, enzymes again change the shape and return to the original size. So now this enzyme again have this is special shape in here, active site, and it's ready to be used again in these chemical reactions. So enzymes, they are not changing. Enzymes can change shape, but they're not dividing or adding to the molecules anything. How it was one protein, it's the same protein will be. And it always will be reused again in this chemical reaction. But during these chemical reactions, enzyme will change substrate. In this particular example, it's breaking down in the steps 3 enzyme breaking down connection between atoms in this substrate and create two products. But in different chemical reactions, what can happen that enzymes sometimes they have different they have two separate active site, and during, like in here, if we have one red enzyme and they have two active sites, one is green for one substrate and one is blue in here active site for the second substrate. And in the next step, what can happen when enzyme on the next step will change shape conformation. For example, it will change shape like that. And in this case, we will have in here blue molecule substrate and in here green molecule. They become so close after this enzyme change shape. So what happening? It could be production of the new chemicals bonds between these two separate substrate, and in the end, one product could be released. So this will be our product, and it's one. So enzymes could be used during catabolic reaction, like in this example, or by synthesis, anabolic reaction to synthesize one product from the two substances. But always enzymes, they do not change. They always will be reused again and again in the chemical reactions. And usually in the nature, in order to transform one chemical, for example, we have rock, some kind of made up of the one chemical, and in order to change it in the nature, not living organism in the nature, to the different type of the molecule product, for example, yes, so we have one type of the molecule, it's substrate, not in the living organism, in the nature. And in order to change it and convert it to the different, absolutely some product molecule, what can happen? In the nature, it's need to be or very high energy, some kind of, for example, thunderstorm will hit this rock and it will melt it and conform in the different chemical reaction. Or this rock need to be under the high pressure. And maybe it will go down under the water and something can happen. Or this substrate need to be fall down in the volcano and be under the very high temperature. And under this condition, it can be converted to the different chemicals product. So this is what happening in the nature. In order to convert one chemical to the other chemical, need to be some kind of very extreme conditions, and which is not often and very fast could happen in the nature, but in our body every second happens several different chemical reactions very fast and very efficient, and conversion of the one substance to the second substance occur all the time. And our body doing this without these extreme conditions such as high temperature or high pressure or something else like that, because we have this enzyme, this specialized enzyme, which reducing, how it's called, activation energy to modify one substance, like in here purple molecule, and make from this different molecule, like in here product red, make it absolutely different. So we use in our cells enzymes, in here show this in the blue. Enzymes is used to do this work, and I already explained mechanism of action, how it's done by the changing shape of the enzyme. And because of this, our body doesn't need this extreme kind of conditions. Without this extreme condition, enzymes catalyze this chemical reaction, increase speed of this chemical reaction without these extreme conditions. So now we will talk about one type of the metabolism, degradation, digestion, or catabolism. And we will talk about catabolism or catabolic reaction of carbohydrates in the animal cells. We will look at an example of hydrolysis. So remember, I was talking about this. Hydro mean water, lysis mean break. So this is catabolic reaction with help of the water also, but of course enzymes. And in order to break down sugar, sucrose, our regular sugar, into the glucose and fructose, our body use special enzyme. So our cells have enzyme, and this enzyme called sucrase. So overall, ACE, it means enzyme. This is what it means. This is end of this. But sucr in here, this is usually mean what this particular enzyme metabolize. And in our case, sucrase, this enzyme will use for metabolism of sucrose. So this is why they have this is similarity in the name. So again, this sucrase will have special 3D shape in here, active site, and during metabolism, chemical reactions, what will happening? Sucrose molecule, which is polymer of glucose and fructose units, what's shown in here. There's glucose and fructose, they have chemical connection between each other. So this is, they have chemical bond. So this is sucrose, will be attached. So sucrose will be attached to this enzyme, sucrase, in here. Key and lock mechanism, precise shape will be this active site of this enzyme. So what next happening? Enzyme will change shape conformation, and because water available everywhere in our cells, so water molecule will be used to break down kind of this connection. So this is what shown in here, how water molecule break down this connection between glucose and fructose units inside of the sucrose molecule. And in the end, what will happening that we will have two separate products right now. We have glucose and we have fructose. And how you can see when chemical bond is broken between in here glucose and fructose in this sucrose molecule, part of the water such as hydrogen ions will be attached to the fructose and hydroxyl unit will be attached to the glucose in here. So this is what happening during hydrolysis of the sucrose with help of the enzyme sucrase and water molecule. And this is when we bridge bond, chemical bond connection between glucose and fructose. And two separate units, glucose and fructose, are released from enzymes. And in the end, what happening? Enzyme sucrase already ready again for next molecule of the sucrose to be attached. So it could be reused all the time. Our substrate, sucrose in here in this case, will be changed during this chemical reaction and two products will be produced, glucose and fructose. Hydrolysis occurs in the digestive system in each individual cell and inside of the cellular organelle, lysosome. Here one of the example, we will talk about enzyme called catalase. So ACE again shows that this is our enzyme, but it's not enzyme which metabolize ketones, of course. So catalase is enzyme actually. It's an antioxidant enzyme which neutralizes hydrogen peroxide. And hydrogen peroxide is an oxidant molecule. So catalase can neutralize this oxidant molecule inside of our cell. When we have accumulation, a lot of this hydrogen peroxide molecule accumulate in the large amount inside of the cell, it's toxic for the cells. And because they're very high reactive, and they cause oxidation and damage of the biomolecules. They're making holes inside of the biomolecules and damaging cells. So catalase is very special, again, antioxidant enzymes which help our cells to remove extra amount of the hydrogen peroxide. Now we will see in here shows this is the large molecule show. This is molecular model of the enzyme catalase. And this is show This is the active site of this enzyme, active site. And our substrate in this reaction will be hydrogen peroxide. This is very tiny molecule, and it will be attaching to this active site as a key lock mechanism, which is showing here. Key lock mechanism, we see this is enzyme, this is substrate, and you see that active site have very unique shape, that only one type of the molecule with exactly same shape can fit in here precisely, like a key and lock. So after this, catalase again will change the shape, and this substrate, in our case hydrogen peroxide, will be catalyzed and break down on the two molecules, actually three, because will be two water molecules in here show from the two hydrogen peroxide molecules, and one oxygen molecule will be released. And oxygen will be used, for example, in mitochondria to produce energy, could be. And water, our cells can use. It's a neutral molecule. It's not will damage our cells. So again, this is how our body neutralize and remove this extra amount of the hydrogen peroxide, which is toxic in the large amount, with help of the catalase, catabolic enzyme in here, antioxidant enzyme. And in here it show also in here diagram how this all done. Other chemical reactions which are happening during metabolism is biosynthesis. So we will look on the biosynthesis of the proteins in the animal cells. And this building synthesis of the new molecules happening during dehydration reaction. So de mean removing, hydra mean water. So this is what happening during this reaction. So we will look what happens. So again, we will have specialized enzymes, and these enzymes will have again active sites with specialized 3D shape for the basic units to attach to this. So in this case, we will have two substrate. We have substrate A, for example, and substrate B. And this substrate will be attached each to their active site in here on enzymes, and they will produce enzyme substrate complex. Next, what will happening? Enzyme will change shape, conformation, and during this change of the shape, these two units will be very close. And water molecule in here, from both of these end of the connections, bonds, chemical bonds, will be detached right now in form of the water molecule. And new chemical bonds will be formed between these two units, A and B. After this, so this is what will form, will be our product, and it will be released. So this is our product, one, released. And enzyme again will be used again for the same kind of chemical reaction. So again, this is an example of the synthesis, for example, proteins. So in this case, we will have amino acids A and amino acids B. And during this enzymatic chemical reaction, we will produce product, which will be protein. So this is simplified, of course, but the point is that during biosynthesis, chemical reactions happening with help of the enzymes, and it's dehydration reaction. So this is what happening in here. We're removing water molecule away when we're connecting to units. Synthesis occurs in each individual cell of the body, and almost all cellular organelles in different ways involved in this process. Biosynthesis of the ATP. So we will see. So again, will be enzymatic process. It means enzyme, particularly it's called ATP synthetase. This name of the enzyme of this will be used to biosynthesis or synthesize ATP molecule. So synthase, ACE mean enzyme, syntha mean produce, and it's produce what? ATP. So this is what name of this enzyme telling us. So this enzyme is enzyme which help to produce ATP molecule. So how is this happening? So we have two active sites in this enzyme, one active site in here red for the ADP molecule, adenosine diphosphate. It have only two phosphate group. And the other active site on this enzyme will be in here green, and it will be for this substrate. And this substrate could be different molecules, but they all have, this is on the end, they have phosphorus group. So this substrate have phosphorus group. So when enzyme will change conformation, what will happen is that this phosphate group will be attached to the adenine diphosphate, and what will happen? And in this case, we will have ATP molecule now because we will have T3 phosphate group, and we will produce new biomolecule from substrate. So this will be product. Yes, so this substrate was round molecule, how they represent. And now we produce different molecules, so it have different shapes, so they represent a square shape. And in here show, in the bottom image, in here show adenosine diphosphate molecule with, it have biosynthesis reaction with phosphate. In here, phosphate, this is ADP. We have phosphate, and what happening during, so usually during biosynthesis, in order to connect this, we will need energy to use, but we will also, because it's enzyme called ATP synthetase, so we will produce also energy in here, ATP molecule.